All right, guys. Mic check. Mic check. Mic check. 1 2 1 2. All right, guys. You go ahead and close your MacBooks for me. You can put up the stuff for your foldables. You will have time after lecture to get that done, so don't stress about it. Don't worry about it. You'll still get a chance to get that done today. So I need you to close your MacBooks, and you should be on page 120 in your note packets. So close your MacBooks, get on page 120 for me in your note packets. 120. Alright, guys and girls and guys, let's get ready to science. Let's get ready to science, guys. Alright, so we are in Unit 5. Unit 5 is a very short unit because it is a continuation of Unit 4. In Unit 4, we talked about cell organelles with cell structure and function. And one of the main organelles that we discussed in Unit 4 was the plasma membrane or the cell membrane. Now, what is the purpose of the cell membrane? It controls what enters and exits the cell, and by doing so, it helps the cell to maintain what we call homeostasis. Now, homeostasis is a tendency to move towards equilibrium to maintain a stable environment. So the plasma membrane helps to maintain homeostasis for a cell by being semi-permeable in controlling what it allows to enter and exit. So the word I just said was semi-permeable. What does semi-permeable mean? Let's break it down. Semi means what? Half. Alright? So that means it's not allowing everything. It's allowing about half of the stuff that tries to get in and out to go in and out, right? And permeable means like to permit. So it only allows certain things to go in and out of the cell. So let's see what actually, what's the thing that makes up the majority of the cell membrane? Which one of the macromolecules, the carbs, lipids, proteins, nucleic acids, we discussed this in Unit 3, which one its job is to make up the cell membrane? Which lipid, specifically? The phospholipid. All right, the phospholipid. So since we know it's a lipid, we know it contains carbon, hydrogen, and oxygen, but because it is the phospholipid, it also contains what? Phosphorus, right. And that phosphorus is found in the phosphate group. So when we look at the structure of a phospholipid, so this is the way you normally see it drawn, but what you actually have is, let me draw this way. You actually have a 3-carbon sugar. Attached to one of those carbons is a phosphate group. No chop. Yes, phosphate is PO4. So phosphate is phosphorus and 4 oxygens. Phosphorus is the element by itself. But when you say phosphate, you're talking about a phosphorus that is bonded to 4 oxygens, okay? And you also have, coming off of 2 of the carbons, you have these fatty acid chains, okay? Now these fatty acid chains are composed of carbon and hydrogen. Of course, this is not a completely accurate picture, but, and we tend to associate the backbone and the phosphate group as being a part of the head of a phospholipid. So when you look at the actual structure of a phospholipid, you can actually label the parts. What about what?
The last carbon, doesn't it have a tail?
No, it has a phosphate group.
What is the
If it would have had 3 tails, 3 fatty acids, it wouldn't be a phospholipid, it would be a triglyceride. Okay? So that's the difference between triglyceride and phospholipid. Triglyceride has 3 fatty acid chains, whereas with the phospholipid, one of those fatty acid chains has been replaced with the phosphate group. Now, so when we label this, we know that this part right here is my phosphate. Okay? That part of the phospholipid is the phosphate. This part right here is my glycerol. Glycerol is a 3-carbon alcohol, okay? So we have our glycerol, and then we have these fatty acid chains. So this last part would be our fatty acid chains. Now, we know that fatty acids can be either saturated or they can be unsaturated. So if I have a fatty acid chain, let me draw these a little bit better than what I did. If I have a fatty acid chain that is composed of every carbon in the group only having single bonds. Remember, we talked about the fact that when you see a structure like this at each one of these junctions, I need to recognize that that is a carbon. And since carbon has to make 4 bonds, if I count how many bonds the carbon is making, I have to also understand that what I don't see are hydrogens, okay? So a fatty acid chain is composed of carbons and hydrogens. If each carbon is making a single bond to each other, then I say that this is a saturated fatty acid, because every carbon in the chain is making a single bond to the next carbon. However, if I have a chain where at least one of the carbons is making a double bond, doesn't have to be many, if at least one of the carbons in that chain is making a double bond, this double bond makes this become what we call an unsaturated fatty acid. Now, they can be mono-unsaturated, meaning they only have one double bond, or they can be poly-unsaturated, meaning that there's more than one double bond. Now, with a phospholipid, this is just an example, okay? You may have where one chain is saturated, the other one is not, or both are saturated, or both are unsaturated. It just really depends on the composition of the phospholipid. The hair keeps getting in my face. All right, so what we have here is the 3 parts. What's this called?
Phosphate.
And what's this?
Glycerol.
So the phosphate and glycerol make up the head of the phospholipid. And what are these things?
The triacid.
Fatty acids. And fatty acids can be what or what?
Saturated or unsaturated.
Saturated or unsaturated. What makes one be unsaturated? A double bond between the carbons, okay? So if every carbon in the chain is making a single bond, then we call that what?
Saturated.
Quick question from Unit 3. Plants or animals, which one naturally makes unsaturated fats? Which one? Plants. Because unsaturated fats are what at room temperature, liquid or solid? Liquid. And saturated fats are solid at room temperature, and saturated fats tend to come from animals. All right, now let's look back at our phospholipid in a so I'm gonna draw it this way this time. What do these little legs represent? The little tails represent the what? Fatty acids. And what do I know is a part of this head? Glycerol and phosphate. Now, phosphate is what we call a polyatomic ion, okay? Phosphate is a polyatomic ion, which means it carries a charge, all right? So that part is charged. Here, with our fatty acids, because they're composed mostly of carbon-hydrogen bonds, carbon and hydrogen bonds are what we call non-polar covalent bonds. That means they share electrons equally. There's an equal sharing of the electrons, and since the sharing is equal, this molecule, this part, these bonds do not have a charge, okay? So they don't carry a charge. So when I look at a phospholipid, this molecule is what we call amphipathic. And what that means is one part of the molecule, one end of the molecule, the charge is different than the other end. Here I have a negative charge on one end and no charge on the other end. Are you with me? cell?
Water.
Water. Water is a polar molecule, okay? Water is polar. What's the chemical formula for water, guys?
H2O.
H2O. Alright, so, drawing 3 elements here. Putting my oxygen here, my 2 hydrogens here. Now, the bond made between oxygen and hydrogen is also a covalent bond, but it's a different type of covalent bond, okay? They share electrons, but oxygen is what we call more electronegative. It has a stronger pull on electrons than hydrogen does. So they share, but it's shared unequally. Because of the unequal sharing of electrons in water, we say that water is a polar molecule. So water is polar. And what we mean by polar is that it has poles. It has opposite ends. For instance, because oxygen is more electronegative, it appears that the oxygen carries a negative charge, because it pulls the electron closer to its center than the hydrogen does. Hydrogen strong, not letting the electron go, but the electron is closer to the oxygen than it is the hydrogens. Because of that, the hydrogens appear to have a positive charge. Do you notice something similar about the ends of this molecule and the ends of this molecule? So water will be considered what? What's that word? Amphipathic. Because one end of the water molecule appears to be negative, and the other end of the water molecule appears to be positive. So on its ends, it has different charges. Here we have negative and neutral, and here we have negative and positive. Now water likes things that are charged like it. Okay? Like dissolves like. So water will interact with anything that carries a charge. And things that carry a charge are polar molecules and ionic molecules. Or ionic, doesn't have to be a molecule, it could just be an ion. Anything that carries a charge, water will interact with. Okay? So, when we look at the phospholipid, will water interact with it?
Yeah.
Yes.
And
Half and half.
half and half. Yeah, it'll interact with one half, but it won't interact with the other half. Why?
Because one is a negative and one is a positive.
Not-
Neutral.
One's neutral, right. Half of the molecule is charged, like water likes. The other half of the molecule is neutral, like water does not like. So what we call this is the half of the molecule that is charged, we say it is hydrophilic. So this half is hydrophilic. What does hydro mean? Water. Philic is from philios, which means loving. So this half of the molecule is water loving. This half over here, hydrophobic. It's hydrophobic or water fearing because it does not carry a charge. Okay? So this half is hydrophobic. So when we look at a phospholipid, the head loves water, but the tails don't. Makes sense, right? Because the tails are just like what we find in fats and oils. Does oil and water mix? No. Oils are hydrophobic. So the fatty acid chains, the tails, are hydrophobic. Now, let's go back to this cell over here. My wonderful, wonderful drawing of a cell. Because I am such a great artist.
Yeah.
I'm going to take over Miss Shrammick's art class. She don't even know it yet. It's coming. Alright, so, don't tell I said it though. I want to be a surprise. So let's blow up this section of our cell membrane. Alright, so the cell membrane separates the internal contents of the cell from the external contents of the cell, right? The main component of the cell membrane is the phospholipid. But here's a problem with the phospholipid. Only half of it likes water. And if it's separating the inside from the outside, we have a problem because the inside is mostly water, and the outside is mostly
Water.
water. You with me, Eli? You sure? Look like you're in another world. Alright, so in order to fix this problem, the phospholipid, when it is in an aqueous solution, when it is in water, it forms a bilayer. What does bilayer mean?
A double layer.
2 layers is a double layer. So what it looks like is they will arrange in the plasma membrane, when it's making the membrane, they arrange tail to tail. So there's 2 layers. There's an internal layer, which goes to the inside, which the heads face the inside of the membrane, and there's an external layer where the heads face the external environment. So it forms what's called a phospholipid bilayer. Okay? By doing so, it keeps the heads that like water interacting with the water, and the tails that do not like water are protected from the aqueous environment. They're secure on the inside. Now, if I look at my cell membrane then, that means that my cell membrane has regions. The region where the heads are located is a hydrophilic region. But what about this region right in here? What about the region where the tails are located? What do I call that region? Hydrophobic. So this region is hydrophobic.
What if
All right.
that double layer gets destroyed?
Then the cell is destroyed.
Oh, then he
Uh-huh.
dies.
Huh?
Then he dies.
The cell dies. Apoptosis. Cell death. Programmed cell death. Alright, now, so this is the main component of the cell membrane, but not the only component. Before we move on to talking about what else is in the cell membrane, let's review what we have up here. What's the main component of the cell membrane again?
Phospholipid.
Phospholipid. What are the 3 parts that make up a phospholipid?
Glucose, phosphate, and acid.
Alright, so it's glycerol,
Fatty acid.
glycerol, phosphate, and fatty acids. Fatty acids can be what or what?
Saturated or unsaturated.
What's the difference?
Saturated has one and unsaturated has double bond between the things.
Carbons. The thingy things are carbons. Alright. Now, what do I know about the phospholipid because of its structure? What is it? What's the word? It's an A word. What does it mean? But that's is what?
Amphipathic.
And what does amphipathic mean?
Half is water and half.
Right. And so with the phospholipid, we know half of it is what?
Like
Polar, and the other half is neutral. So we have a charged half and we have a neutral half. And what do we know about water? It's like things that's like it. So water is also amphipathic because one end of the molecule is negative and the other end is positive. Which element carries the negative charge? Oxygens. And which elements carry the positive charge? The hydrogens. Alright, so water will interact with only half of a phospholipid. So which half does it interact with? The tails or the head? The head. Now, with a cell, the majority of the inside of the cell is what?
Water.
And the majority of the outside of the cell is? So these are aqueous environments. Okay, aqua, like aquafina. Aqua means water. Aqueous environment. So the environment is mostly water. So how does the phospholipid?
Double bilayer.
It forms a bilayer or a double membrane. Which part of the bilayer is facing the inside and outside?
The hydrophilic is in
The heads. And the tails are where?
outside of the head.
In the inside of the membrane. And so that means that the region on the inside is what? Of the double membrane, of the cell membrane, of the bilayer, the region in the inside of the bilayer, what is it? How is it classified? Hydrophobic, and the outsides of that double membrane are classified as hydrophilic. Cool beans, y'all killing it. It's awesome.
I have a question.
What's up?
What's neutral? What do you mean by neutral?
No charge.
Oh, so negative.
No, negative is a charge.
Zero.
0. Yeah, if it's negative or positive, that's a charge. Neutral is no charge. Okay, so 0. Alright, now let's talk about the membrane itself.
Oh, I get it now.
You got it now?
So water goes to positive and negative, but doesn't go to zeros.
Okay. Yeah, if that's how, yeah. If that's how you got it, that's how you, yeah.
Awesome,
Alright, so the majority of a cell membrane, we know, of course, is the phospholipid, right?
Yes.
Alright, so let's, now, the reason why I drew some of those that way is because whenever there's a double bond or an unsaturated fatty acid, it makes a kink in the chain. The chain is no longer straight. The double bond causes the direction of the chain to change, okay? Now, that's the equivalent of saying, putting your arm out and saying, keep your distance. So if you were walking in a line, or you were in a straight line, and everybody had, or if you're standing shoulder to shoulder, everybody have their arms down, right? So you could start, people could start moving closer and closer and closer to you, right? And you can go from where you had some room to move around to where you packed in tight. Y'all with me? So cell membranes need to have room for the phospholipids to move. They move side to side, okay? They move laterally. They need to have room to move. That's called fluidity, okay? Its ability to move is called fluidity. Now, what happens to molecules when the temperature gets cold? What do?
They start to freeze.
They start getting closer together, right? That's a problem for the cell membrane. That's called losing the fluidity. And the problem is, if the membrane gets packed too tight, nothing can get in or get out. So the cell membrane can't do its job. So that means no waste products coming out and no nutrients coming in, okay? So that's a problem. So saturated and unsaturated fats, the composition differs by animals and plants that live in different climates. I'll get you in 1 second. Live in different climates because of the temperature. So if an animal or plant is accustomed to being in a colder temp, they're going to have more unsaturated fatty acids, which don't allow phospholipids to get too close. So the purpose of unsaturated fatty acids is to help the membrane keep its fluidity. Like you moving closer, it's like, nah, bro, 6 feet COVID. All right, what's your question?
If they get too close, like they can't get pushed away, will the cell die?
Yes,
So ice, it's a ice cell thing.
exactly.
So ice is good for you?
And depending on how cold it is. Alright. Alright, nothing of anything. Too much of anything is bad, right? So too much of anything is bad. So now, the purpose of unsaturated fats is kind of like I said, it's kind of like putting your arm out like, whoa, cuz, getting too close, keep your distance, 6 feet. Alright, there's another molecule that can also be found in the cell membrane that does a similar thing. And that molecule is made up of 4 fused carbon rings. It only contains carbon, hydrogen, and oxygen. It is the basis of making steroid hormones. Which molecule is that? Cholesterol. It's a lipid, but it's the lipid, specifically cholesterol. Okay? So cholesterol can also be a part of a cell membrane. And the purpose of cholesterol, which is another type of lipid, is to keep the cell membrane fluid as the temperature changes. Now, the unsaturated fatty acids only work in cold weather. But cholesterol works in cold or hot. So if it gets really, really cold or really, really hot, the cholesterol helps the membrane to keep its fluidity. So that's not the only thing that you can find extra in a cell membrane. All right? You can also find proteins. All right? Proteins can be embedded in or attached to a cell membrane. Sometimes a protein only goes part of the way through the membrane. So it has a job on one side of the membrane. or the other. This hair is killing me. Sometimes a protein goes all the way through the membrane. So it goes from one side of the membrane to the other. And then sometimes a protein may just be loosely attached to the membrane on either side. Now I want you to notice something. Do you notice how even though I have a bilayer, the composition of one side can be different than the composition of the other side?
Yeah.
That's important, okay? Because the inside composition of the bilayer is for the inside of the cell. The external composition is for the outside of the cell. So they can be different. Now, quick question about Unit 4. Which cell organelle produced the proteins, transported the proteins, and made sure the proteins made it to become a part of the cell membrane? There's 3 organelles you need to tell me about. I need to know which one produced the proteins, transported the proteins, and packaged the proteins and make sure they made it to the cell membrane. What are those? I heard ribosomes. What else? Tell me. Golgi and the rough ER. That's it. So that's how they work together. These proteins that are a part of the cell membrane would have been produced by ribosomes on the rough ER, transported by the rough ER to the Golgi, packaged and sorted and packaged by the Golgi and sent to the plasma membrane when they became a part of it. So, do you see how that works together? All right. Now, let's talk about why this is even a thing in the first place. So, we said the job of the cell membrane is to control what goes in and what goes out of the cell, right? And we know that this cell membrane has regions. We know that this region right here is hydro what? Philic. All right, so we have a hydrophilic region, and that's going to be the same here and here. So those are hydrophilic regions. And we know that this region in the middle is hydrophobic. Now, here's the thing. In order to simply cross the membrane, something has to be both small and hydrophobic. So, a compound that is small and hydrophobic, something such as oxygen, is able to sneak between the cracks in the heads. That's why the heads need to be able to move. That fluidity allows little spaces in between the heads. So it has to be small enough to sneak between the cracks because that area is hydrophilic. But it comes to this big area right here that's hydrophobic, and like dissolves like. So the only way it can make it across this big region is it has to also be hydrophobic. So anything that can freely cross the membrane has to be both small and hydrophobic. That's an and, not an or. Now, if I take something that's larger, let's say something like a glucose molecule, C6H12O6. And when I say small and large, look how many elements. There's only two atoms that make up an oxygen molecule. But when I look at a glucose molecule, there's 24 atoms. So this is a lot bigger than that one, right?
24.
24. 6 plus 12 plus 6.
That was 2.
Now, if this tried to simply cross the membrane, it couldn't fit through the cracks. It would get rejected. So it instead has to find another way in, and that other way is through a protein. Proteins create channels, or they carry molecules across the membrane. A channel is just like an open door, okay? A channel is an open door, but it's still specific. It only allows certain things to come through it, all right? A carrier is like a door that actually has to open and close. You attach to one side and it opens up to let you go through to the other side. So the proteins can be channels or carriers. Now, this one was non-polar. It doesn't have a charge on it, does it?
No.
It's non-polar, but it's large. And you have to be what? Small and non-polar. I can take something like, let's say calcium ion. It's only one atom, Well, one ion, which is just an atom that has lost or gained electrons. So it's small, but guess what?
It's hydrophilic.
Still can't go through because it's hydrophilic. So it has to be small and nonpolar. So it has to find another way to cross the membrane. So this is what I want you to remember. If something is large or charged, okay? Large or charged. This means it's hydrophilic. Doesn't have to be both. It can be large. If it's large, it has a problem. If it's hydrophilic or carries a charge, it has a problem, because things that are hydrophilic are polar or ionic, and polar and ionic substances carry a charge. Okay? So, what does it have to be to go through the membrane? Small and
Neutral.
right, hydrophobic, non-polar, neutral, no charge. Make sure you associate the word hydrophobic with non-polar or neutral. Alright? Now, if it is not those two things, then how will it cross the membrane?
Protein.
Through a protein channel. Alright, now, also, we tend to see on the outside of the membrane a lot, we tend to see another substance that is made up of carbon, hydrogen, and oxygen that we typically associate with a source of short-term energy storage. And that substance can be attached either to, let me put a protein up there, either to phospholipids or proteins in the membrane. That substance is a what? A carbohydrate. Carbohydrates attach to proteins or lipids on the plasma membrane, and it's used for cell-cell recognition. The reason your immune system knows what cells in your body are the cells that they should not attack is because of the composition of the carbohydrates that's on the external membrane. When it attaches to a lipid, we call it a glycolipid. Glyco, not Geico like the insurance. Glycolipid. If it attaches to a protein, we call it a glycoprotein. But your immune system knows which glycolipids and which glycoproteins should be on which cells in your body. It should know, unless you have an immune system like mine that decided it wanted to attack itself. But that's a whole nother, that's called autoimmunity. When your body gets mad at itself and like, I'm going to kick my own butt. All right. So anyway, the purpose of carbohydrates on the plasma membrane is cell recognition. It allows a cell to recognize a like type. A skin cell knows a skin cell. Ain't that right, Cam?
Yeah.
A skin cell knows a skin cell because of the carbohydrates that's on the surface. Okay?
So the carbohydrates are like dots. They know everything.
No, we ain't gonna go that far. But they like wearing an ID tag.
Yeah.
They're like IDs. All right. Now, let's turn to practice 1 on page 1. We don't have to turn this on page 120. Let's do practice 1 on page 120. Any questions about anything we went over today?
Yeah.
What's up?
Okay. I have a question. Okay. Can I see this? Okay.
Use your fingers.
Okay. Okay. So this is the head.
Mm-hmm.
The head. Okay. And these you said that this is inside the cell membrane.
Right.
Okay. So what is this, inside or out?
If this is the inside of the cell, then this is called the internal face of the membrane. This whole thing together is the cell membrane. Okay? So the cell membrane has an internal face, an external face, and then just the inside. So this is the inside of the cell membrane. If this is the outside of the cell, then this is the external face of the cell membrane. And if this is the inside of the cell, then this is the internal face of the cell membrane. It's like if I did this, right? If this was a membrane, see how I got a part in the middle? That's the inside, but then I still have the 2 edges, right? So heads, tails in the middle. Got it? All right.
Okay. And DC also said that it's not a good thing, like if it's too cold or one thing is not too good, right?
Mhm.
Yeah. So money's not good.
Too much of it is not, it corrupts. Go sit down.
Give it to me, curiosity.
I'm a teacher. Where do you get money from? Alright, I'm about to ask you for something. Alright, anyway. So, let's go to page 120, practice 1. Muhammad, since you're so enthusiastic today, go ahead and tell us what A is.
It's the head.
I know it's the head, but I want you to tell me if it's hydrophobic, hydrophilic, polar, or
It
non-polar.
likes water.
So what does that mean?
Hydrophobic.
Phobia is fear.
Follic.
Alright, hydrophilic, and that also means it's what? Polar or non-polar?
Polar.
Polar. Alright, so hydrophilic and polar. That's A.
Lick.
Shania, because I know I just saw you pick up that phone. Bring it to. Bring it on. Bring it on. Bring it on. Bring it on. And while you're coming, look at your paper before you walk up here so you can tell me what B is.
Steady.
I'm gonna bring it with me.
Uh-huh. Uh-huh. Gracias.
No fucking more.
Alright, so what is B?
Fatty acids.
Hush, I'm talking to Shania. I'm talking to Shania. And that ain't even an else. Alright, so what's B?
Fatty acids.
Hush!
Oh.
Then that's calling up the wrong else. What's B?
See,
Uh-huh. See what happens when you be on your phone doing class? Makaya, help her out. What's B? Hydrophobic. So that means, is it non-polar or polar?
Non-polar.
Non-polar. Alright, now, real Eli. Oh, will you stop the recording for me? So I don't have to edit out when we call people names since they don't want to put that online like that.
It's okay to put my name.
I have to bag it up. Thank you.